Welcome grade 12s. Today we're going to be looking at DNA, the code of life. It's my favorite part of the grade 12 syllabus. Can be quite easy. You just need to listen very carefully and listen to the words that I use. Okay, and if you look very carefully at the board, this to me, this is one of the most important parts. And you think to yourself, why would this checklist be important? It's really important. It's telling you exactly what do I need to know. So after you've studied, all right, can you answer these questions? Do I know this? Am I able to describe this? Am I able to do that? All right, so when we come to DNA, right, we are looking at something called nucleic acids. And when we look at DNA, the most important thing, all right, that you need to be able to do, all right, is to know that we're going to find two different types of nucleic acids. And you're going to see over here, oopsie, let's get that going over there. All right, name the two nucleotides. Let me get my pen going. Oh, I love pink. Right. Name the two nucleotides. Already, the nucleic acids, you must be thinking RNA. You must be thinking DNA. In your mind, I need you to know, what do they look like? If I were to draw them, what would they look like? RNA is single-stranded. DNA is double-stranded, and it's twisted into this double helix. Okay? They're like Lego blocks. What are they made up of? They're made up of nucleotides. And when I talk about a Lego block, what am I talking about? I'm talking about a sugar. I'm talking about a phosphate. And I'm talking about a nitrogenous base. That is my one Lego block. And that block, if I put them together, right, I'm going to make this really big structure called a nucleic acid. I obviously can't call it a Lego block. I'm going to call it a nucleotide. All right, we go back. Where do we find them? We go back to our grade 10 work. In the cell, what do I need to know? I've got a cell. The cell has got a nucleus. Inside that nucleus, what am I going to find? I'm going to find my DNA, my chromatin network. My DNA, all right, unwinds and becomes familiar to me as chromosomes. All right, where do we see that? Again, we see it in meiosis. We're going to see it again when we look at sexual reproduction. Okay, so it's a concept that's going to follow through. Outside of the nucleus, I have cytoplasm. All right, and in the cytoplasm, I have got these organelles called ribosomes, okay, called ribosomes. Those are going to be my main features, all right, when I come to looking at the DNA. Because what am I going to be looking at? Why do I need to know it? I need to know the structure of DNA, right? Because my DNA is going to be involved in certain, all right, procedures that are essential to the cell. The DNA is going to be involved in replication, making a copy of itself. Where am I going to find that important? Meiosis. Where else am I going to find it? Mitosis, right? Looking back. Chromosomes. My DNA unwinds. What do I see? Chromosomes. What are on those chromosomes? Genes. Where do I find that? I'm going to see that in my genetic section, and also when I get to my sexual reproduction. So the start of it has a lot, all right? It links into each other, all right, into the different parts that you're going to look at. Okay, so for the DNA, as I said to you, I need to know the structure. I need to know what's different between RNA, what's different between a DNA. How did I get RNA, all right? Those kind of things. Where do I find it? So the how, the where, the why, very important. Okay? You need to be able to describe processes like DNA replication, transcription, translation. These are all very important words that you need to be able, all right, to discuss, to be able to recognize. When I talk about RNA, RNA comes in different forms. Okay, I'm going to see something that's called mRNA, which is messenger RNA. I'm going to see a structure that's slightly different called tRNA or transfer RNA. Okay, they are different and they're going to have different roles in the different processes. Then maybe the more tricky part of that is the two words transcription and translation. Okay, transcription is to make RNA from DNA, and then translation is to take that RNA and to make amino acids join together to make a protein. And why do we say protein is so, why is this process so important? Remember, the proteins that are made, most of them are life-giving, they're essential. Hemoglobin, without it, we can't absorb oxygen, we can't breathe. We need to make insulin. Insulin is a protein, right, that helps with our glucose content. Again, you're going to do that when you do the endocrine system, right? What else? Very important when it comes to making blood clotting, right? Hemophiliacs don't have that protein, so their blood doesn't clot. So it's life-important processes that we're going to look at. So when you look at your checklist, it's important, do I know these? Because if you know it, then you'll be able to answer the questions. Now I'm going to go through a few questions where I've taken those essential elements and we're going to look through them. Okay, the first one, all right, is actually a multiple choice question. There are 3 of them relating to one diagram. Now, if you have a look, it says the following diagram outlines the production of proteins in a cell when DNA is activated. So it's already told you, all right, what are we doing? Sorry, we are making proteins, and that is called translation. All right, this diagram in front of you now, you must become very familiar with it. Right? It's a diagram. Certain diagrams like this appear in almost all of, all right, your papers. Now, if you have a look very carefully, you will see, if you have a look over here, let's quickly, here's DNA. All right, there's my DNA. Then, what does it say? Look here. I've got a process here that goes out the nuclear pore, and there is another strand, which we should already start seeing that it's mRNA. Okay, and where does translation occur? On that organelle called a ribosome. Okay, so now, what do we need to know when it comes to the translation, the transcription, etc.? The most important thing that you always need to remember is the complementary basis. The complementary basis. What do I mean by that? Remember in my nucleotide, all right, if I draw, I've got a sugar, I've got a phosphate, and I've got a nitrogenous base. All right, and always remember, nitrogenous base goes to sugar, all right, because if you have sugar, something's always going to attach to your hips. Can you see here? Sugar is always attached with something, and that's your nitrogenous base. Now remember, these nitrogenous bases have got partners, all right? They always go together with their partners. The word there, complementary basis. Now remember, adenine always goes with thymine, and cytosine always goes with guanine. Okay, that is the rule for DNA. What is the rule for RNA? RNA, we have a little bit of a change. We don't find thymine. Adenine always goes with uracil, okay? And cytosine and guanine, they always go together. They're always nice and faithful to each other. Okay, now the first question can be quite tricky. I want you to pay attention. It says here, at stage 1, the DNA molecule involved has at codons 5, 6, and 7 the base sequence. All right? At stage 1, the DNA molecule involved has codons 5, 6, and 7, the base sequence. Now, if we have a look, and I gave this to my matrics, and this is why I've given it to you, because they get quite, what was tricky is as soon as they saw this question, they honed in on this number 5 over here. They said, okay, that is number 5. That is not codon 5, 6, and 7. That is amino acid number 5. If you have a look at the diagram very clearly, all right, you're going to see it says there, start. And what they're saying there is that's where you start to count. So they want codon 5, 6, and 7. And remember, what is a codon? A codon is a triplet. A codon is a triplet. So we can put over here triplet, okay? Because how many little bases will a triplet have? 3. Okay, so let's count. 1, 2, 3, that is codon 1. 1, 2, 3, that is codon 2. 1, 2, 3, that is codon 3. 1, 2, 3, that is codon 4. 1, 2, 3, that is 5. 1, 2, 3, that is 6. 1, 2, 3, that is 7. Okay. Now, the question, so what are we looking at? We want to know, at stage 1, the DNA molecule involved has at codons 5 and 6 the base sequence of. This over here is my RNA. This is my RNA. I want to know what would the sequence have been on my DNA. Okay, now I quickly want to just go over that because I went over it a little bit too much. Okay, let me go over this one a little bit. There we go. So if we have a look, there's number 1, 2, 3, 4. So number 5, let me just go over that again. Number 5 is G, A, C, all right, G, A, C. Then we have G, let me erase that quickly. A, that's number 6. Then we've got C, C, C. All right, now be careful. You've got the code. They want to know what the DNA would be, all right? Not what the tRNA would be, what the DNA would be. So if I go up a little bit, those are my rules. Those are my rules. A goes with T, adenine goes with thymine, cytosine goes with guanine. G goes with C. A goes with T. T, all right? It's on the DNA. C goes with G, G goes with C, G goes with C, A goes with T, G, G, G. All right, so let's have a look over here. What would the answer be? C, T, G, C, C, T, G, G, G. And my answer would be number C. Very, very important. If you're not sure, all right, if you're not sure, have a look, read your question carefully. It's the terminology, all right, the words, that's what sometimes you get a little bit stuck with. They asked for the codon, the triplet, all right, and they wanted to know what was on the DNA. So, very tricky question, that one. Okay, same diagram again. At stage number 2, all right, here's stage number 2. What they want to know is structure K made of tRNA? All right, let's have a look. If we have a look carefully over here, all right, where is structure number K? Here. Structure number K, what is it? It's a ribosome. All right, remember it's my organelle where translation occurs. So is it made of tRNA? No, it's not. All right, my question incorrect. The 3 bases of group H form an anticodon. Here's group H. If this is my codon, what do I always find on my tRNA? Yes, my anticodon. All right, so that would be, there we go. My answer would be number B. Let's just go and have a look to see that I've made sure. Number C, bond J represents a hydrogen bond. All right, we got over here, we see there's a bond. Okay, what bond is that? It's a peptide. If you notice, a lot of the proteins, peptide, or peptide bond, proteins. Okay, so it's not. Where am I going to find a hydrogen bond? In my DNA. So we know that it's definitely not number C. Number D, the mRNA will code of a polypeptide containing 10 amino acids. All right, we can't tell from that. Right, we're not quite sure how many amino acids that it's going to, all right, code for, because it's only a portion, all right, of the diagram has been shown. Okay, let's go on to the last question of this series, question number 1. All right, now the anticodon for glycine. Now you're going to see very often you're given the names of the amino acids. You don't have to study them. The amino acids will always be given to you in a table or here as it is in the diagram. This looks quite tricky, this question, but it's actually not. It's actually as simple, all right, as the question actually looks like it's going to be. It says the anticodon for glycine. So number 5, you'll see there is glycine. And if we look at the diagram, there is number 5, all right? And what do we know? Is amino acid, it is attached to a tRNA. I say transfer, but I often refer to it as a taxi because that's exactly what a taxi does. It takes things to and from, and that's exactly what it does with amino acid. Picks up the amino acids, takes it to a place, loads it off, goes back again, picks up another amino acid to go back again. All right, so the anticodon for glycine is, and there we go, and what is attached at the end of a tRNA? An anticodon. Right, and there it is. Tells you there's the anticodon, CCU. Right, the answer is number B. Okay, now let's take a look for the next question, question number 2. You're going to see a very similar diagram. You're going to notice that the diagrams are quite similar, so it should show you kind of pattern, right, of the questions that they're going to ask. Okay, I start off with a very simple question first. Okay, this is a more theoretical question, and then going on, so it's the beginning part, question number 2, and then I'm going to show you how we take this, and then we move it to question number 3. All right, have a look over here. It says, the proportions of the 4 organic bases in a piece of DNA from a squirrel, all right, a piece of DNA from a shark, and a piece of human RNA are given below. Okay, now it says here, now have a look over here, be very careful. The squirrel, I'm going to use the 2 S's. The squirrel and the shark, it has their DNA. All right, DNA is different to RNA. Remember why? It has got a double helix and it has thymine as one of its bases. Whereas the humans, all right, we've given as RNA, so it's a single strand, right, and we're going to find uracil as its base. Okay, now let's see if we can answer the questions. If you notice from the start, I need you to start thinking. A squirrel, a shark, and a human. 3 very different organisms. But what do you know? Nearly all living organisms have got the same basis. They might be totally different, right? But they all have the 4 bases. There we go. Similarity, similarity. So if they've all got the 4 bases, why are they different? Let that start to mull over in your head a bit. Okay, let's look at question number 1. This is quite a trickish question, but again, as I said, it's a bit of a theory question. You must just tie it up to when I spoke about in the beginning, complementary basis. Okay, question 2.1, it says, explain why the proportion of A and G is approximately equal to the proportion of T and C in both pieces of DNA. Huh? Right, what they're saying. It's a bit of a difficult question. Again, let's read it through carefully. Explain why the proportion of A to G, all right, is equal to the proportion of T and C in both pieces of DNA. All right, now what did I say to you? What is DNA? First thing you need to know, DNA is a double strand. Right? And what do we know about the double-stranded DNA? Very important. Complementary basis. Now let's have a look at the table over here. What do we mean by when we look at DNA? Oopsie daisy. All right, if we look at A, all right, A, there's 29, shark, there's 28, all right, and the human, we're going to just looking at the DNA. Okay, A, what do we know? A goes with T. 29, 28. Similar. Shark here, 28, 29. All right. So if we're looking at it, we're looking at a proportion of 1 to 1. In other words, there are as many A's as there are T's. There are as many C's as there are G's. So the complementary basis, A and T and C and G, why is there the same number? Because, all right, they have to be the same number because they need to join to each other. It's a double strand, all right? So if I've got an A on one side, I have to have a T on the other. So explain why the portion, all right, the proportion. The DNA is double-stranded, it's 2. It has complementary bases, therefore adenine must always have the same as thymine, and cytosine must always have the same as guanine, so that the DNA can join. It's a double strand, all right? It's a double strand. How can you have one on one side and not a complementary on the other? That is the whole secret behind the DNA. If I've got one on one side, I need to have its complementary base on the other. So my numbers are always going to be equal. Okay, now question 2, similar question, but slightly different. Now it says, if we have a look here, explain why a similar equality does not exist, all right, between the proportion of A and G and the proportion of U and C in the mRNA. Have a look over here. 40, 15. Okay, 15, 30. Remember what I said here. If we have a look, 21, 22, almost exactly. 22, 21, exact. So why on the mRNA do we not have the same proportion? What is the mRNA? The mRNA is a single. Yes, it doesn't match up. It's a single strand. So because it doesn't match up, all right, with a partner, yes, we made it from DNA, but the mRNA is by itself. It doesn't have to have a proportion, right? Because it's a single strand and it does not have to have equal partners. Okay, let's use the word bases there. Okay, right. Question number 3. Despite being very different organisms, right, the squirrel and the shark have similar proportions of the 4 different bases in their DNA. Explain how this is possible. I said it to you in the beginning. All living organisms, okay, have, let me say, I'm not going to say all, that would be weird to say. Living organisms have the same bases. So the squirrel and the shark all have A, C, G, and T. All right? So despite being very different, right, why, explain how, all right, even though they have the same in similar portions, how are they different? They have the same basis, but what do they have? It's very simple. But in different sequences. All right, next time you guys go to the shop and you go and you buy something and the cashier puts it underneath a scanner, all right, all that it's done, and how come a different price comes up every single time? But they've only used the same 10 numbers, a 0, a 1, a 2, a 3, a 4, a 5, a 6, a 7, 8, 9. And depending on the order in which that barcode, all right, appears, will depend on the price. Okay, same numbers, same thing here, same letters, same nitrogenous bases. But if I put them into a different sequence, I'm going to make a different organism. Okay, your barcode. Okay, that is going to be all for now for question number 2. We're going to take a quick break, and when we come back, we're going to look at question number 3. Welcome back, grade 12s. I hope you had a small little wee break, and now we're going to carry on with the rest of our questions. Okay, we're going to start now with question number 3. As I said to you earlier, this is a diagram, right, that is very common, right, when we get to our DNA section, especially when we're dealing with transcription and translation. Let's read the question carefully. It says, study the diagrammatic representation of the role of DNA. Oopsie, let me go back here to my pen. Oh, pink, all right. It's telling you the role of DNA and RNA, and it tells you during the process of protein synthesis, okay? So it's telling you, we know that the 2 nucleic acids, RNA and DNA, and it's going to be involved in that protein synthesis. When you do that, start already with the terminology, translation. All right, okay. So really you need to have that word going over in your mind. When you look at the diagram, don't get too overwhelmed, all right? Because it looks like it's a lot of information all at the same time, and there's lots of lines and there's lots of letters, right? Slowly, you have a look, go through things that I've already said to you, okay? Remember, my DNA, double-stranded. My RNA, single-stranded. My DNA has got thymine. My RNA has got uracil. Little hints that can show you which of the molecules are involved. Okay, so let's start off with a question, right? And then we'll see, does it look as confusing as some of you might seem to think. Okay, first one, 3.1. Please listen very carefully to the terminology. Identify the cell organelles represented by X and Y. Now, when I'm talking about a cell organelle, I'm talking about a structure, a membrane-bound structure within the cell. Have a look at X. That is the first one. Lots of you, when you see X, the first thing you do is you want to label it DNA, because you see it's this double-stranded. They didn't ask you for the nucleic acid. They asked you for the organelle. In other words, which part of the cell is X? And the X is my nucleus. And I know that why? Because where do I find my DNA? I find my DNA in my nucleus. Okay, so X is nucleus. What is number Y? This is number Y. Where does the mRNA go and rest on? What is that organelles? All right, it is the ribosome. Very careful. As I say to you, identify the cell organelle. That word was very important, because if you just look at it, some of you see, oh, Double strand, oh, that's a nucleus. Why? Single strand, sorry, that's the mRNA. Those are the nucleotides. They asked you for the cell organelles. Those are the structures inside the cell that we drew earlier. Okay, let's go into question number 3.2. Same diagram, obviously. Identify the molecules. Identify the molecules numbered 1, 2, 3. All right, now look here. It says 1, 2, 3, not 1 and number 3. Number 1, number 2, and number 3. Little things like that that can cost you those very small marks. Let's start. Number 1, okay? They asked you, what is this? They ask you what number 1 is. The molecule. The DNA is a molecule. Number 1 is my DNA. What's my number 2? Now here's my number 2. Now look very, very carefully. Number 2, they're pointing to this structure, which is my single molecule. Molecules are large. Number 2 would then be my RNA, not, okay, my RNA. If you want to say mRNA, that is fine. It usually only referred to it as messenger RNA when it goes out the nuclear pore. All right, then number 3, we go all the way down here. Can you have a now look here? Number 3 is this whole structure over here. So now, what is it? It's carrying an amino acid, okay? And attached to it seems to be these 3 bases. What carries an amino acid, all right, and is going to attach to the mRNA? Yes, correct. My 3 molecules are my different nucleic acids. So number 1 was my DNA. Oh, number 2 was my mRNA. Okay, and number 3 was my tRNA. See the difference in questions? They are molecules. The others were organelles. Nice, easy marks if you read the question carefully. Okay, next one, question number 3. Identify the groups of nucleotide bases represented by 7 and 8. All right, there are only 2 groups. Can you understand what we're looking at? Where do we group them? Right, let's have a look. Number 8 is over here. That's number 8. And number 7 is at the top here. Okay. So, number 7, where is the group? The group is on my mRNA. Now, you can use 2 words here when it comes to this one. All right? I like to use the word codon. But it's another word that you can use, all right, is called a triplet. And why it's easier to remember triplet is because, as you would notice, how does the mRNA group itself? Into groups of 3, and triplet means 3. So we've got 3 bases, 3 bases, 3 bases. So triplets or codons, all right? Codons. Now, if you remember codons, then it's easy to remember number 8, because what does a tRNA bring in? What is it carrying? It's carrying the complementary bases to the codons, all right? And they are opposites, and we usually use the word anti for opposite. So that would then be my anticodon. All right, so number 7. Number 7 is the group. Look at my question again. My group of nucleotides. My group. So on my mRNA, I have codons or triplets. On my tRNA, I have all right, I have anticodons. Okay, not anti-triplets, all right? Can't use that word. So triplet might be easier to remember because we know they're in groups of 3, but codon and its corresponding partner, anticodon, go together much easier, okay? Remember, anticodon on my tRNA and my codon on my RNA. Okay. Question number 3.4. Give the name of the process. Name of the process. Okay? During which the molecule number 2 is formed. Now, if we have a look, where is number 2? Okay? Molecule number 2 is mRNA. All right. What is the process called whereby I make mRNA? Remember, DNA is in my nucleus, and I need to make a protein, all right? And DNA is too big, all right? It's too big to go out of the nucleus, so it needs to send a messenger, all right? And the DNA is responsible for coding that message, all right? And we call that process transcription. Okay, transcription, making of RNA, from a DNA. But they asked you to name the process. One name, transcription. Transcription is when I make RNA from DNA. Translation is when I then take that RNA and then make the amino acid. Okay, make the protein from that. Now, those have been quite simple. Now the longer question. All right, now the longer question. Get your thoughts in order before you start writing. They've asked you now to describe the process of translation. Okay, translation is how we make a protein. Now, when you describe the process, it is important that you put it in order, right? You need to put the whole process in order. Now, when you look at it, look at the diagram they've given you, okay? And try and tell a story from what you see. Okay, so how does translation go? Now, if I were to tell the story, okay, translation starts with this. How do I get that? Okay, how did I get to that point? Because I know in transcription, I made the RNA. Okay, the RNA went out the nucleus and it went here, my messenger RNA. It's now sitting on the ribosome. But how do I make a protein? What's the process? This. Let you think about how this starts. Okay, let's have a look. What is involved? What is involved? tRNA. Okay, tRNA is involved. So let's start. I'm going to put it in point form because sometimes it's easier for you to put your thoughts down in point form. You don't have to write all the paragraphs. When it comes to your essay, that is where the paragraph format is important. Here, if you battle, put step by step in a bullet. And it's also easier sometimes for the marker to mark. So let's start off with number 1. What do I have? I have a tRNA present in the cytoplasm. Okay, there we go. That's what I just said. I've got a tRNA. What's attached to the tRNA? The tRNA has anticodons attached. Right, so look here. There we go. There's my anticodon. So I'm looking at the diagram. The diagram says I've got a tRNA. Attached to it is my anticodon. Now, what else is attached to it? Remember, it's almost like a balloon. Here I've got my taxi, all right? And here I've got it 3 little, all right, it's 3 little codes. And what do I go and get? What is the tRNA? It's a taxi, remember. What does it pick up? The tRNA, all right, picks up its complementary amino acid. Okay, have a look, 5 marks. I've already got 3 points. Now it's carrying its amino acid. What does it now do? Where does it go? It attaches to its complementary. Do you see I use that word often? That's very important in this because that's exactly what they are. It's complementary codon on the mRNA. Okay? Now what do we know? Look, I've got an amino acid, amino acid, amino acid, amino acid. I always tell my matrix it's like a kebab. All right? I got a piece of chicken, got a piece of chicken, I got a piece of chicken, each is amino acid. Got a chicken, got a piece of chicken, got a piece of chicken, got a piece of chicken. Now, how am I going to eat all of these chickens at once? It's impossible. So what do? I need to join them all together. Okay? How do I do that? Simple. I take a Sestati stick and I poke them all through. And what have I got? I've got one big molecule, the protein. Okay? So that's exactly what happens here. What do? All right? The amino acids, they lie next to each other. Okay? And then what do? They form a bond. Yes, a peptide bond. Right, and when they form a peptide bond, what happens? There we go, the amino acids join. And when we have 50 or more amino acids, what do we have? A protein. What is that? Translation. Okay, translation. 5 marks. I have got 1, 2, 3, 4, 5, almost 6 points. In order. In order. All right? I pick up my text, I pick up my amino acid, I join, I click in. All right? Where all the amino acids, they lie next to each other, they're all waiting. What do? I join them, I join them, I join them, I join them. What do I make? I make a protein. Protein goes out the way. What does the taxi go and do? What does my transfer RNA go and do? It breaks up, goes back, and it picks up something else to make another protein again. Okay, that is translation. Now, going on from that, you need to become familiar with this table. You don't need to study this table. It will always be given to you. All right? There's a variety of ways in which it can be given, but you do not need to know the amino acids. A table like this will always be given to you. Now pay close attention to the question. It says here, a fraction of an insulin molecule was extracted from cattle. The sequence of the amino acids looks as follows. Okay? All of those. Now, what do you have to do? Write the sequence of the base triplets for the 8 amino acids mentioned above as they would appear on the DNA. All right. Now, it might take you a bit longer, but do it slowly. Okay, have a look here. Let's start. Where do I start? Here. All right, now have a look here. His, all right, is actually histamine. Now look what it says. Histamine is CAC, but look where that's the code. All right, so I'm going to put here CAC. Can you see that? Histamine is CAC. Write it. Leu is leucine, CUA. All right, then cysteine again is U. So let's go cysteine, UGC. Gly is glycine, CGA. Okay, there we go. Serine, AGC. Histine again is CAC. Lou is CUA. And val, all right, is there for valine, and valine is GUA. That is the codon on the mRNA. They give it to you, all right? This is database. All the information is given to you. Now remember, I only need to know the triplets or the base, all right, codons on any one, DNA, mRNA, or tRNA. And from that, I can work out what the code is going to be. Now it says here, write the sequence of the base triplets for the 8 amino acids mentioned above as it would appear on the DNA. Here, so I must take these, all right, and I must now convert it to what the original code was. As soon as I see DNA, all right, as soon as I see DNA, you must think already, okay, no, all right, no uracil, no uracil. Okay, very important. I'm going to use a different color now. I'm going to use green. Okay, so let's start. C goes with G, A goes with T, C goes with G. C, G, U goes with A goes with T. Remember, no uracil, all right? It's on the DNA. The DNA, the rules are simple. A goes with T, C goes with G. Okay, so U is an A, C, G, G, C, T, T, C, G, G, T, G, G, A, T, and G, A, T. All right, sorry. That should be C. Apologize. Let me quickly do that. There we go. C, A, T. 1, 2, 3, 4, 5, 6, 7. How's that for nice 8 easy marks? All right, nice 8 easy marks. And all the information was given it to you. They asked you for the DNA. They could have asked you for the anticodon, but they didn't. Okay, we're going to take another little break, and then I'm going to see you just now. Welcome back, grade 12s. We're going to go into our last section of our exam revision. All right, and we're now going to look at question 4. I'm not going to read the whole paragraph to you, right? But basically, question 4 deals with what we call DNA fingerprinting. And let me give you the scenario quickly. Here was a mom who doesn't know, all right, she has a child and she doesn't know who the father is. Okay, so remember, from DNA, we can actually test paternity, right, to see who the father was. Now, if we have a look, this is the most important thing. If we have a look at a DNA fingerprint, it's like a barcode, all right? And this is the information that was given after the paternity test. This is Ben, who is the child, all right? And what we're trying to see is if Johnny or Zach is the father of the child. If we have a look at question number 1, that's the first question, right? Who is Ben's father, and justify? How do you give evidence? Okay, I want you to go back here. When we look at DNA fingerprinting, this is Ben. Now, it's quite simple. To be related, you need to have as many markers in common. Okay, so let's go. Here's Ben, and there's Johnny and Zach. Okay, that equals Ben has the same as Johnny, okay? Ben has the same as Zach. 2 for 2, okay? Then here, same, same, same. So here it could be any of them, all right? Now what do we notice? There we go, none. But if we look at the end, all right, what do we see? We see that Zach, 1, 2, 3, 4, 5, 6, 1, 2, 3, 4, 5. Zach has got more markers in common than Johnny. So when you use a fingerprint, all right, here, we said here, who's the father? The Zach, and the answer was because he has more markers. All right, so in other words, he has more in common. Now you need to think, the next question over here is give one advantage and one disadvantage of DNA profiling in a paternity test, okay? Why would it be advantageous for a mom to know who the dad was? And the first thing that comes to mind for me is money, okay? And what we're talking about there is maintenance, right? Because usually that is the responsibility of the father to be able to, obviously, right, be able to support his child. What could be the disadvantage of it? A disadvantage, all right, if you think what I came up with, one of the thoughts that I thought is if the father, all right, doesn't want anything to do with the child. Right, so imagine if you know that your father, right, your biological father, might not want anything to do with you, all right, and even though you know paternity, you know your father, he doesn't want anything to do with you, he doesn't want to support you, all right, that could actually be, you could feel neglected. Okay, so come up with any advantage, disadvantage. Now this next question is it's a tricky one. It says here, one can never conclusively prove paternity. One can only disprove paternity. Oh, that's quite a difficult sentence. What do they mean about the scientific truth? Okay, what it means is you can never conclusively prove paternity. In other words, even though Zach, right, had as many markers, right, as Ben did, which proves that they are family, we don't have enough information. They could just be similar markers. Doesn't necessarily mean that he's not his parent. But it's true to say that if they have got no markers in common, then they obviously are not related. Think about genetics. What do we know about genetics? Genetics, you carry your traits from one generation to the other. So if they're the more dissimilar, in other words, the more traits they don't have in common, proves that they are definitely not related. But the fact that they have certain traits in common doesn't necessarily mean that they are. It just means that they could have, we don't have enough information to prove without a doubt. Tricky one over here. Okay, next one, you must know. What else other than paternity, right, can we use DNA fingerprinting for? Remember, criminal cases? All right, I'm sure you all watch CSI and all of those things. Okay, to identify bodies, remains, right, that's how they use during the war and all of those things, when they find bodies and they don't know who they are able to do it. In South Africa, we can use it for poaching. How do we mean by that? We can use DNA, take DNA from animals, rhinos, etc., and if we find the horn, we can positively prove that that is one of the animals that was poached. Okay, all right, now just remember going back there, it says there, other than, you cannot in your answer tell me to see who the father is. It says other than. Okay, then the last one. This is a question where it's your opinion. Your opinion can be yes or no, but the crux of the matter is, can you justify it? Ben is a minor and could not give consent for his DNA profile. Do you think it's ethical that Patsy obtained his profile? And here, all I want from you, not a yes, maybe, yes, no, maybe, is a yes or a no. That's it. One mark. Don't sit on the fence. A yes or a no. Then give reasons to support your answer. If you say no, you could have said it invades his privacy. All right, it might hurt him. You must then justify the reasons why you said no. Or you said yes, because the father must do right by the child, right? He must pay maintenance. He must look after the child. The child has a right to know who his father is. The child has a right to know where his origins are. Okay, so when it comes here, it says, do you think it's ethical? Your answer is a simple yes or a simple no. Don't say maybe, Okay, that's your 1 mark. Then it says, give a reason to support your answer. It must be a logical reason, not just because. Okay, yes, as he has a, it is ethical. Why? Because he needs to know who his father is. The mother is looking after his best intentions. No, it's an invasion of his privacy. When he's older and he understands, maybe then we can do it. All right. Thank you for tuning in, grade 12s. Have a wonderful day and enjoy the rest of grade 12. We are almost finished, and good luck.